I think we've learned more recently about co-alterations with KRAS and how they may impact how patients do, specifically with chemo IO, but I think we've even learned at AACR, at least on a virtual presentation this year, with single agent IO. How do we use other alterations to make treatment decisions? We've learned, I guess I'll just start out saying we've learned that potentially if you have a co-alterating STK11 mutation or a KEAP1 mutation with KRAS, maybe those patients don't benefit with adding immunotherapy to chemo, although we're still learning. And maybe it's different if you have a P53 with KRAS, those patients do better. What are your thoughts on this? And do you use this routinely to make decisions?
I don't think it's something that I would do in a standard of care fashion. But I think oncologists are used to using all the data they have in front of them, both the clinical data, the biochemical data, for instance, chemistries, what's the renal function. And so if you have data in front of you where it might be helpful, very often, to me, there's not a right and a wrong answer. There are several options. So I do take them into account, but it's harder to deselect. I'm not going to offer Mrs. Jones this treatment because she has an STK11. I don't think we're there yet. And in fact, some of the analyses of large trials have been contradictory, saying that KEAP1 or STK11 was not predictive, that it was prognostic. Another gene of emerging importance here is ARID1A, chromatin remodeling gene, and we'll be quite a large series at ASCO this year, virtual, on the impact of that biomarker, which was reported to be predictive of outcome. 11 and KEAP1 were not and missed it. Now, I'll have to make a confession, Ben. I got time zones mixed up in my calendar. And when I tuned in to virtual AACR yesterday, I was 3 hours off and it was the lung session was ending. So tell me what you heard at AACR that I should be interested in. I may have seen it on Twitter.
Yeah, no, that's why we're doing this, so we can catch each other up. My understanding, and I haven't read the full abstract yet. I saw the Twitter feed push looking at Keynote 024, a small subset of patients looking specifically at those who had STK11 to see if that blunted the effect of immunotherapy over chemotherapy for this patient population greater than 50%, and it didn't. It didn't. It seemed the results seemed to hold up. And I'm sorry if I'm butchering this data. I'll look at it again. But that's my understanding based on a very small subset, that STK11 didn't seem to knock out the effect for patients that were PD-L1 greater than 50 treated with Pembro versus chemo. And I think we just need more data to better understand the relevance of these co-alterations. You've mentioned others that are emerging, which again speaks to the importance of doing comprehensive genomic profiling so that you can get the full story and not just chapter 1 of a tumor.
Right, I agree. Nothing is ever as simple as it seems at the beginning.